prokaryotic gene regulation, kind of the stuff we talked about last class. And understanding how different operators, repressors, and activators work will allow scientists to alter bacteria to suit our needs. So last class we talked all about operators, repressors, and activators. Why do we care? Why do you need to know this stuff? Well, think about a new field called synthetic biology. It's a brand new area of research that combines science and engineering, and scientists are making new life forms to do work not normally found in nature. Why do you care about this stuff? Well, think of the best scientists we have today. We have people like Craig Venter. Here he is on the cover of Time magazine. He led Celera in the sequencing of the human genome back in the early 2000s. He partnered up with the Nobel Prize winner Hamilton Smith, who discovered restriction enzymes. They started the J. Craig Venter Institute, and they wanted to create synthetic life. And for their discovery, for creating synthetic life, they were on Time magazines. They were one of the best inventions of 2008. Can you believe it? You create a synthetic organism, and you're just number 21 on the greatest inventions, right? So the number one invention that year was the retail DNA test. So amazing, amazing invention. Why do you want to do synthetic biology? Why do you want to create new life? Well, imagine creating bacteria that grow and fluoresce in the presence of oil. So if there's an oil spill, it's kind of hard to detect, especially at night, especially if you have small particles of oil. Well, imagine if you have a bacteria that grows, that fluoresces wherever there's oil. Imagine not only that, you get that bacteria to produce oil-degrading enzymes, just like the tripopron, lacopron. If you see oil, turn on activators, turn off repressors, turn on oil-destroying enzymes, right? That's amazing for cleaning up the environment. As well, you can generate bacteria that produce clean-burning oil for you to use. So let me show you just a very quick video showing some of that clean-burning oil.
You know, I'm trying to find a good place on the slide where...
That's it, stop. This round thing is a blob of pure diesel oil.
So these things here are the bacteria. So see the rod shape, there you go.
Yeah, right, there's one.
They're in the midst or just finished converting renewable sugar to secreted ultra-clean diesel to an oil product.
Feed sugar to these modified bacteria cells, and they give you diesel oil.
That big blob there is simply oil. That's our ultra-clean diesel product. It is a finished product. We have to separate it, wash it a little bit, and it's ready to go.
Yeah, I mean, you're really blasé about this, but that is completely nuts. You know, you've got microbes producing diesel.
We're really excited about it, Bill. We've been working really hard on this.
Yeah.
I think the microbes have been working harder.
This is revolutionary stuff. And Steve and his team have set up a test refinery where these manipulated bacteria are now producing not just a few drops of oil, but gallons of it. And this is what you end up with. In this case, pure diesel. And just in case you think this is a hoax, watch this.
Start to understand the power of these types of things, right? We're talking about simple operators, simple activators, simple repressors, but by exploiting this type of technology, we're doing synthetic biology. Okay, so this class we'll talk a little bit about synthetic biology, but we want to start off by just reviewing those simple activators and those repressors. So here we have a recap of prokaryotic gene regulation. This thing's very simple to read. You just basically read it like a table. So this thing is basically a table. So on the top, you have negative regulation. Negative regulation is when the bound repressor protein prevents transcription. You want to alter this slightly because it's a little bit confusing when you're studying. So write down when bound to DNA. Right? So when bound to DNA, and then the rest of it's fine. So when bound to DNA, when bound to DNA, the repressor protein prevents transcription. So negative regulation. When bound to DNA, that repressor protein prevents transcription. Same thing over here, positive regulation. Adjust your notes a little bit. When bound to DNA, that'll make it easier for you to study. When bound to DNA, the activator protein promotes transcription. Okay, so you have your two columns, negative regulation, positive regulation. Not only do you consider what the protein does, though, you have to consider what the ligand does. So the ligand could be a nutrient, could be a waste, could be some type of small molecule, could be some type of peptide, whatever. Okay, so ligand could be a nutrient, could be a waste, could be a small molecule, could be sugar, could be oil, right? Some type of thing. Nutrient, waste, small molecule, peptide, whatever. Okay, so not only do you need to consider the big protein if it's negative or positive, if it's a repressor activator, you also need to consider the ligand. So if the ligand binds, well, it might remove the protein from DNA. Sometimes ligand binding will allow the regulatory protein to bind to DNA. Okay, so we'll go over this one step at a time. Up here in this corner, we have negative regulation. When bound to DNA, the repressor protein prevents transcription. So when it's on DNA, you prevent transcription, but you also need to consider the ligand. So the ligand binding here removes it from DNA. So we're saying without the ligand, you bind to DNA, the gene is on. With the ligand, you remove it from DNA, so the gene turns on. We talked about an example of this last class, right? This would be the LAC repressor. That's one example. Allolactose takes it off of the DNA. All right, so let's consider the option down here. So again, it's still negative regulation, as in when bound to DNA, this repressor protein prevents transcription. You also need to consider the ligand in this case. Here, the ligand binds to allow the regulatory protein to bind to DNA. So with the ligand, the repressor protein binds to DNA, and the gene will be on. If you remove the ligand, well, the gene will be on, right? And so an example of this would be something like the TRP repressor. With 2 molecules of tryptophan, the TRP repressor would bind to its DNA. Another category we have here is positive regulation. So again, when bound to DNA, the activator protein promotes transcription. Here, ligand binding removes it from DNA. So activator protein without a ligand binds to DNA, helps RNA polymerase bind, gene is on. If you have a ligand, you take that thing off of the DNA. Last option's down here. So again, same thing. When bound to DNA, the activator protein promotes transcription. However, ligand binding in this case will allow the protein to bind to DNA. So with the ligand, this thing will now bind to DNA. That will help RNA polymerase bind and get the gene on. If you remove the ligand, well, you'll shut it off. So example of this is CAT, the catabolite activator protein. All right, so these are basic examples of prokaryotic gene regulation. When you go through it, make sure you know all these like the back of your hand, right? Because you're going to have to map this out on exams and stuff like that. These are really good examples. We did not talk about an example of this top one. That's okay. You still might see it on the exam. And remember, with the LAC repressor and CAP, it's a little bit more complicated, right? Because we talked about how 2 conditions must be true last class, right? So you can go back and take a look at that. We also talked about the tryptophan operon last class, and we talked about how the tryptophan repressor contains a helix-turn-helix motif. All of you wrote this down, like I watched you write this down last class, right? So you have the slide from last class. The helix-turn-helix motif is shown here. So the tryptophan repressor, part of the protein has this particular shape, where part of it is a helix, then it turns and has another helix. And if you have that shape, generally that lets you fit into the major group of DNA. Some proteins, a lot of different proteins may have this shape, and in general, if you have this shape, you can probably fit into the major group. And different amino acids will be there to recognize different sequences. But the tryptophan repressor has one that allows it to bind to the specific sequences of the tryptophan operon, the operator there. And we said last class, 2 molecules of tryptophan, 1 red one here, 1 red one here. That's going to allow the tryptophan repressor to change in conformation and allow it to fit into the major groove quite nicely. So one major groove fitting right here, another major groove fitting right here. Right? So you guys all wrote this down last class, you're kind of good to go here. Recap. Negative regulation is competition between RNA polymerase and the repressor protein for promoter binding. With positive regulation, an activator protein recruits RNA polymerase to the promoter and activates transcription. Again, this whole section is about how you can't really change your DNA, but you can change what comes out of it. Two ways to change it. You have negative regulation and positive. Negative is when you have competition between RNA polymerase and the repressor protein. If a repressor protein is there, RNA polymerase can't bind. And positive regulation is where there's an activator protein. It's actively pulling down RNA polymerase and say start transcription. Right? So two methods of transcriptional regulation. Now, gene regulatory elements are typically very close to the transcriptional start site of prokaryotic genes. This is an example we talked about last class. You saw this exact picture last class where you have a start site for RNA synthesis, LAC-Z, so that's going to be the LAC operon. You have the RNA polymerase binding sites, including where the sigma factor binds, the operator, the cap binding site, right? So that's all from last class. Now, normally, especially in bacteria and prokaryotes, these things are very close to the transcriptional start site, but not always. Regulatory elements can also be found far upstream, as in far before where the transcription starts. Can be downstream in eukaryotes, as in after the gene. Could also be within genes, could be in the introns. Again, those sequences that activators and repressor proteins bind, they can be far upstream, as in way, way before the transcriptional start site. They can be downstream of the gene, especially in eukaryotes. They can be after the gene. They can also be within genes, as in introns. And of course, that's going to be eukaryotes, right? Prokaryotes don't have introns. So a lot of people think, well, introns, they're not coding, they're not important. No, that's not true at all, right? You can have regulatory elements there. You can have stuff that activators and repressors bind there. Some regulatory elements are distant from the transcriptional start site. Again, some are way before, some are after, some are in introns. How do you control transcription? Well, DNA can loop. One example is with DNA looping and what's called NTRC. NTRC, as it says, is a transcriptional activator. So let's zoom up on this figure right here. Okay, zooming up. Here is the DNA. Here is the promoter. So you have bacterial RNA polymerase. The sigma factor could be there, but they don't show it to you. Here is NTRC. It's binding to a sequence that's really far away. That's way upstream of this gene, right? The gene's way down here. How is this NTRC that's binding so far going to affect the RNA polymerase? Well, DNA can loop, so it's going to loop the DNA, come over here, and I didn't make this up, they say it gives the RNA polymerase a kiss, right? So it kisses the RNA polymerase, and now the gene goes on, right? And you can transcribe it and do the thing here. So this is the electron micrograph of what it looks like. So you can imagine the DNA is the line through here. This big circle is one of the proteins. This big circle is another of the proteins. You can see how the DNA kind of loops there. So RNA polymerase can be activated from a distance via these types of DNA looping mechanisms. Yeah, that is the recap of prokaryotic gene regulation. So we're going to start to talk about how transcription can be controlled via different circuits. We'll see where I'm going with this. And the first thing we want to talk about is bacteriophage lambda. And that is confusing right off the bat because students will see bacteriophage lambda and they'll say, oh, that's a bacteria, right? No, it's not a bacteria, it's a virus. So bacteriophage lambda, I know it says bacteria, but it is a virus. And it is, in fact, as it says here, a virus that infects bacterial cells. So you see bacteriophage lambda, you go, nope, not a bacteria. Virus that infects bacterial cells. All right, so take a look at the figure right here. This is your bacterial cell. So you look at this and you know, oh, that's not bacteriophage lambda, right? That's a bacteria. So here is the bacteria, here is the host chromosome, here is the lambda virus. So this is bacteriophage lambda right here, bacteriophage lambda. Green will be the protein, red is the DNA. So you have a protein shell, you have DNA. Bacteriophage lambda will, as it says, attach to the host cell and inject the lambda DNA. So it is infecting this bacteria. The lambda DNA will circularize. So there's other enzymes involved. You don't have to know the other enzymes for this course. But there's other enzymes involved. The DNA will circularize. From here, what we're going to see is that positive and negative regulatory mechanisms are going to work together, and they're actually going to repress each other's synthesis. Okay, quick review gets more complicated. Bacterial cell, not bacteriophage lambda. Here's the host chromosome. This is bacteriophage lambda here. It's going to inject its DNA to infect the bacteria. The DNA will circularize. From there, there are two options. The first option is under favorable bacterial growth conditions. So imagine the bacteria is in favorable conditions. It's going to grow very happily. So what happens is the virus is going to integrate its lambda DNA into the host chromosome. So the DNA of the virus gets in there. Lots of enzymes involved. You don't have to worry about those enzymes. So here is the viral DNA in red. Now, when the bacteria replicates, it's going to replicate its DNA. It doesn't know the difference between viral DNA and regular bacterial DNA. Right? DNA is DNA. It's just A's, G's, C's, T's. It's double-stranded. It's just DNA. Bacterial DNA polymerase, you see an A, it's going to put a G, right? So you see an A, you put a T. You see a C, you put a G, right? It doesn't know the difference. It's just going to replicate the DNA. So as the bacteria grows and as the bacteria replicates its DNA, the viral DNA is there along for the ride. It doesn't have to do anything, right? Bacterial DNA polymerase doesn't know the difference. It's just going to replicate the DNA. So what's happening here is the virus is essentially using the bacteria as a house, right? It's just hiding away, living happily. Every time the bacteria replicates, hey, the virus gets a free ride. It replicates too. Now let's see what happens when the host cell is damaged. So when the host cell is damaged, we have what's called an induction event. So that means host cell damage. And the specific damage in this case is DNA damage. So induction event, think DNA damage. All right, virus is living very happily in here. It's going along for the ride. Now the bacteria is getting damaged. The house is crumbling, right? So the house is crumbling. The virus says, I'm going to get out of here. So what happens is the virus will get out of there. You get the synthesis of viral proteins needed for the formation of new viruses, rapid replication, lots of viral proteins, lots of viral DNA. It's going to make new viruses. There's so many viruses in here, you're going to explode with viruses. These viruses are going to find a new house, right? Lytic pathway. So those are essentially the two lifestyles of the bacteria, or of the virus. And we're going to talk about two gene regulatory proteins responsible for initiating this switch. Okay, so I just want to review it very, very quickly because it gets very confusing. Again, bacteria gets infected by a lambda virus. Here's the lambda DNA. Two states. Under favorable growth conditions, viral DNA goes in, it's along for the ride. Anytime the bacteria replicates, viral DNA goes along for the ride, right? Bacterial DNA polymerase doesn't know the difference. If you have an induction event, if you have DNA damage, what happens is the virus says, my house is crumbling, I'm going to get out of here. So you're going to exit, virus is going to make a lot of new viruses, and this cell will essentially burst with viruses. These viruses will find a new home. So you have here the prophage pathway as in viruses just sitting there happily, and the lytic pathway bursting with viruses trying to find a new house. Prophage versus lytic. So, two proteins are involved here. You have the lambda repressor protein and the CRO protein. So I'm going to explain to you these two figures. You don't have to jot down the notes here. You have the notes over the next two slides. I'm going to show you more notes. So it's essentially just from your notes. Now, in the first state, we have what's called the prophage pathway. So you remember the virus is going to live happily in the bacteria. Here is the viral DNA, or part of the viral DNA, right? And it's going to be in the bacterial DNA. So here's part of the viral DNA. The CI gene will get transcribed to RNA. It will get translated to what's called the lambda repressor. And the lambda repressor is a repressor. So it goes over here, binds to the operator, and shuts off CRO. During the prophage state, you have lambda repressor shutting off CRO. Now for the lytic state, when the virus is escaping and leaving and making lots of viruses, what happens is instead CRO gets transcribed, CRO gets translated, and this CRO protein binds to the operator and prevents the lambda repressor, right? So they kind of duke it out, right? One inhibits the other inhibits the first one. So what you have here, lambda repressor occupies the operator, blocks the synthesis of CRO, activates its own synthesis, and most bacteriophage DNA here is not transcribed. So same thing that I just said, this is the prophage state, so the virus is living happily in the bacteria. Here's part of the viral DNA. So CI gets transcribed, CI gets translated, you have this lambda repressor. As it says here, as you wrote down, the lambda repressor occupies the operator, blocks the synthesis of CRO, shown right here, right? CRO is off. Most bacteriophage DNA is not transcribed. There's a little special nuance to this, as in lambda repressor, not only does it block the synthesis of CRO, it actually also functions as an activator. So it represses CRO, but it activates its own synthesis, as in it pulls down RNA polymerase and says, transcribe more of this, transcribe more of this CI. Okay, so again, it blocks the synthesis of CRO, it also activates its own synthesis. It pulls down RNA polymerase, says make more lambda repressor. Okay, so that's lambda repressor. CRO is, of course, kind of the opposite, right? It blocks the synthesis of the repressor, allows its own synthesis. Most bacteriophage DNA here is extensively transcribed. So we have here the lytic state. The lambda CRO protein is being made, so you have the CRO gene being transcribed, being translated. CRO comes back here. As you wrote down, it blocks the synthesis of the repressor, so the lambda repressor is off. Now be careful here. CRO is a little bit different than the lambda repressor. CRO is not really an activator. CRO is just purely repressor. So CRO is going to repress lambda, and it just kind of allows its own synthesis. It's not going to actively pull down RNA polymerase. And in the lytic state, most bacteriophage DNA is extensively transcribed. You're going to make those viral proteins. DNA is replicated, packaged, new bacteriophage is released by a host cell lysis. So, just kind of as a summary, what triggers the switch between the prophage and the lytic state? Well, it's the host response to DNA damage that inactivates the repressor. So again, the induction event, that's DNA damage, and if you have DNA damage, what we see is that the repressor is going to be inactivated. No repressor means lots of the other one. So under good growth conditions, the repressor protein turns off CRO and activates itself. It's going to be a positive feedback loop. It maintains the prophage state, and this is an example of what we call a transcriptional circuit. So remember, activates itself, well, so CRO doesn't quite activate itself, right? It just allows its own synthesis. So be careful. So CRO will activate itself kind of just by allowing its own synthesis. And this is, be a little bit careful here. These are two separate points, right? So under good growth conditions, the repressor protein turns off, right? CRO is a little bit different. So take a look at this again, just emphasizing it for you one more time. Lambda repressor blocks CRO, also activates its own synthesis. And then CRO here, it blocks the lambda repressor and kind of allows its own synthesis, not quite activating RNA polymerase here. Okay, so what do we mean by transcriptional circuits? Here are a couple of examples of transcriptional circuits. Be a little careful here because don't forget, this is transcription. There's other types of circuits. There's protein-protein interactions. There's post-translational stuff. All sorts of other circuits. This is transcription. So for transcription, one example is positive feedback loop where A gets transcribed and gets translated. So here's the A protein. And we're talking about a transcriptional circuit. So this A protein goes back to the DNA and increases more transcription of A, right? It's a transcriptional circuit. Over here is a negative feedback loop, so A will get transcribed and gets translated. This circle is the A protein, and it's a transcriptional circuit, so the A protein goes back to the DNA and says, don't transcribe anymore A. Shut it down. You have a flip-flop device, the example we talked about, the CRO repressor, that's an example of these flip-flop devices. So if A gets transcribed and translated, you will have A protein. A protein would go to B and stop B from being transcribed. If B gets transcribed and translated, here you have B protein. Well, B would go to the A DNA here and stop A from being transcribed and translated, or transcribed at least. You also have a feed-forward loop where A gets transcribed and translated, so here's the A protein. A protein would go towards the B area, the B sequences, and stop B from being transcribed. And if you had, would facilitate from B from being transcribed and translated. If you have B, well, B would move forward to Z here and get more transcription of Z, and Z would eventually be translated. A here feeds forward, as in A skips B and goes straight to Z and allows Z to be transcribed and eventually translated. So we'll talk about this one in a second. Now, don't forget the main point here. I think this is fairly self-explanatory if you just look at the diagram, right? But don't forget, it's a transcriptional circuit, as in the proteins here, they're not binding to other proteins, they're going back to the DNA, they're affecting transcription, right? They're affecting transcription. And the repressor protein, right, so not CRO, but the repressor protein, remember, activates its own synthesis, as in lots of repressor protein goes back to the DNA, pulls down RNA polymerase, and you get more transcription of the repressor. Okay, so the first one we want to talk about is this positive feedback loop. Students will look at this and go, oh, that's really simple, that's really boring, right? I get it. A gets transcribed, A gets translated, you get lots of A protein. A protein goes back, affects more transcription of A. Big deal, who cares? That simple system is used to create cell memory. This is how your cell remembers stuff. So let's look at a quick example here. Here's a parental cell. Imagine you start off with no A, right? So if there's no A doesn't get transcribed, nothing's happening here. A transient signal here turns on the expression of gene A. So maybe there's the right ligand. Maybe there's a nutrient there, maybe there's a waste there, maybe the cell ate a piece of bacon, right? Doesn't really matter. There's some type of signal there that says, okay, you better make some A. After you get that signal, well, you have A protein. So A protein goes back to the DNA and you'll transcribe more A. So the cell remembers, oh, I have A. Now, when this cell divides into two, proteins get into both cells, right? So this cell's going to get a bit of A, and the cell over here will get a bit of A. And so if you have A in this cell, it's going to make more A, right? It's a transcriptional circuit. And the cell down here, it will have a bit of A, so it's going to make more A, transcriptional circuits. And as these cells keep on dividing and keep on dividing, all their cells will have A. So all the daughter cells, all the great-great-great-granddaughter cells will remember way back in the day we got that transient signal. They will remember. And that's incredible, right? So imagine you remember, one of your cells remembering, oh, my great-great-great-grandmother ate a piece of bacon, right? That's insane. And so we'll talk a little bit more about this. So you don't have to write this next part down, but we'll talk a little bit about more about this in the second part of the course when we talk about stem cells. So say an egg gets fertilized by sperm. Well, it's a blob of a cell, right? It's a stem cell. Eventually, some of the stem cells change to something like neurons, like brain cells. But you don't have to keep on telling that neuron, you're a neuron, you're a neuron, you're a neuron, right? The neuron doesn't have to remember, I'm a brain cell, I'm a brain cell, I'm a brain cell, right? You don't have to remember that. You just need this transient signal right at the beginning. You change into a brain cell, well, it's going to keep those proteins around that keep it a brain cell. So it will remember it was told to be a brain cell. You don't need to keep on telling it that signal. So that's what we'll talk about in the second part of the course, but that's a transcriptional circuit. Okay. So the second example we want to talk about is a feed-forward loop. Feed-forward loops can measure the duration of a signal. Here, both A and B are required for transcription of Z. So input, here is the DNA, right? So input affects transcription of A. Eventually, you get a lot of A protein. Well, A protein can affect B, can also affect Z. So some of the A protein will move forward to start affecting Z. Some of the A protein starts affecting B, so you get transcription of B. Eventually, you get translation of B. And once you have B protein and A protein both together, now you get Z protein, right? You get transcriptional effects of Z, and eventually you get the Z protein. That affects output. So what happens here is that with a very brief input, you actually will not get enough B, and Z will not be transcribed. Okay, so if you look at the graph here, what they're showing you is on the y-axis, 0 means nothing and 1 means on. So 0 is off, 1 is on. And the x-axis is just on. So if you look at the input here, if you have a very brief input, right, so it's off, it's off, it's off, and eventually it's on just for a bit, you just have a very, very brief input, well, maybe you'll get some A, but you don't have enough A to activate down here and have enough A to activate B. So with a very, very brief input, you're not getting enough B, and so Z will not be transcribed. You won't have enough B to do this thing. Now here you are with a longer input, so imagine for a long time you get lots and lots of input. So if you look over here, lots and lots of input, activating lots and lots of A, you have lots of A being transcribed, eventually you have lots of A protein. Well, if there's enough A around, some will go towards Z, and some will go towards activating B, and finally you have enough B, and then Z will be transcribed. So it helps you modulate the sensitivities. Now, this is actually a very difficult thing for students to understand in terms of the sensitivity, so I just want to give you kind of a real-life, everyday example. And don't scratch up your notes. I'm just going to show you here. You don't need to scratch up your notes. So imagine the input is someone bothering you, and I say imagine the output is you punching them in the face. Okay? So don't punch anyone in the face, but that's the example. So let's say you have this case right here. So you do not have B. It's just A going towards Z. So someone bothers you just a bit, you get A, you punch them in the face, right? So you're out of control. Your friends are like, oh, he has no chill. You're like, temper's out of control. No one loves you, right? You're so imagine on the other side, you don't have the feed forward, right? So you don't have the feed forward. First case was too sensitive. This case right here, not sensitive enough. So someone bothers you, you're transcribing more of A. Someone bothers you, getting more of A. Eventually you have enough A, someone bothers you some more. You have enough A to start activating B. B's being transcribed, right? So they're bothering you even more. You're getting more of B being transcribed. Finally, you have enough B, then you punch them in the face, right? So now they're picking on you, right? So now you're not sensitive enough. But with this kind of feed-forward device, you're kind of at the moderate range, right? So someone's bothering you, right? You're transcribing a bit of A, some of A moves forward, maybe you get your fist ready, right? Someone bothers you some more, get enough A, eventually you activate B, right? And now you kind of punch them in the face. So it's kind of the medium ground, right? So that's where that sensitivity lies. So that's why we have these types of feed-forward devices. Okay. So, transcriptional circuits. All those different transcriptional circuits, you get huge combinations of them in eukaryotic cells. We'll talk about lots of these types of transcriptional circuits, especially in the second part of the course. You get extremely complicated networks. Scientists can construct artificial circuits and examine their behavior in what we call synthetic biology. So synthetic biology, that's exactly what we talked about in the preamble, right? You're using these things, you're going to create your own synthetic network, your own synthetic pathway. One example is creating a simple gene oscillator using what's called a delayed negative feedback circuit. So oscillator, just something that maybe goes up and down at regular intervals. So up and down, or on and off at regular intervals. It oscillates, it changes at regular intervals. Like, so regular you can use it like a clock, right? Oscillates. Okay, so this is, so I know it says here, so this example is where scientists wanted to create a simple system, just something that goes on and off at regular intervals, using what's called a delayed negative feedback circuit. So students are like, well, why do you want to create this, right? Why do you care that you created something that goes on and off at regular intervals? It's just what we call a proof of principle experiment, as in there are all these different parts. You just want to engineer something so that you can prove to yourself that you understand the different elements, right? So set up a goal, make up something, try to engineer it, make sure you understand the system. So just something to try to test out to make sure you understand everything. These are the different parts. I'll go through how it works. Let me just summarize the parts for you. A, B, and C are three different repressors. Lac repressor, we talked about that previously. The TET repressor, which responds to an antibiotic. C is the Lambda repressor. Main point here, three different repressors. Here is the binding site for repressor C. Here would be the coding sequence for A. Here is repressor A, the protein. This would be the binding site for repressor A. Here is the coding sequence for B. Here's the B protein. Here's the binding site for B. Here is the coding sequence for C. So, and this would be the C protein. Different elements kind of all arranged in this type of a circuit. Now, before I show you how this actually worked, is that they engineered this system and they placed this circuit into bacterial cells and watched it work. So take this whole thing, put it into bacteria, you hope to see genes go on and off at regular intervals. You hope to see genes oscillate. Okay, so it's in bacterial cells. And as it says here, so this was a Nature publication. As some of you may know, Nature, Cell and Science are our foremost journal. So if you're a scientist, if you get published in Nature, Cell, or Science, that's a really big deal. Just for comparison, I have never been published in Nature, Cell, or Science, so it's a really big deal. as it says here, we use 3 transcriptional repressor systems that are not part of any natural biological clock to build an oscillating network, right? So these 3 things, all from different species, they're all over the place. We engineer them, or they engineer them together to create a gene oscillator. So how does it actually work? Well, what's going on here? So let's say we start off with A being expressed. So if you have A being expressed, right, so from the DNA you get A being transcribed, you get A being translated. Here is the repressor protein A, right? A is a repressor protein. So if you have A would bind to this binding site, and it's a repressor. So it's going to shut down B, so B is going to be repressed. Okay, so we said we started off with A being expressed. You have the A protein, A is a repressor. So A goes to the DNA over here and will shut down any expression of B. If there's no B around, well, that means nothing is repressing C, so C is going to be expressed. If you have C protein, well, C is going to go back over here and eventually shut down A. So you can see A is starting to oscillate, right? It was on, and now it's going to turn off. And so this is what they're showing you here. Again, on the y-axis, 0 means off, and 1 means on. This is time. So if you look at A expression, well, A starts off being on, but eventually what's going to happen is that it's going to be shut off. And that's what they show you on the next slide. So if you just look at the next slide, you can see A is now being shut off. So A will be repressed, then B will be expressed, and C will be repressed. Okay, that's a little bit more complicated. Let's just follow me one more time. Right, follow me one more time from the last slide. So we said we started off with A being expressed. You have A. A goes to the DNA here, and it's a repressor. So it says, let's shut down B. No more B. So B is repressed. If no B is binding here, that means C is going to be expressed. So if you have C, C will come over here, and eventually will shut down A. And that's what you have on the second slide, right? No more A. If there's no more A, that means, oh, now we can make B. So B will be expressed. And B will come over here and shut down C. If there's no C, well, you get A, right? So it's going to oscillate on and on, on and on. This is a very simple gene oscillator. It's what we call delayed negative feedback, as in, you know, back here, A is repressed, eventually gets expressed, eventually gets repressed, but it's a delayed negative feedback. It takes a lot of steps before you repress A again. What they predicted would happen was this. So imagine, it doesn't matter which one, let's say red is protein A. So they predicted that A protein would go up and down and up and down and up and down. What they actually saw was this. So if you look at one fluorescently tagged protein, this is what you see. Looking at one fluorescently tagged protein. This is in fact what you see, right? AES, it goes up and down, but look, the amplitude is getting higher, right? You're getting more A in total. And that's because they put this oscillator in bacteria, and bacteria grow. You get more and more bacteria. So in total, there's going to be more A as time goes on. So these guys got published in Nature, I have not, and they forgot that bacteria grow, right? Okay. So that's one example of delayed negative feedback. So make sure you do review it. Review it again in the third hour if you don't quite understand it. Another example of delayed negative feedback is in circadian rhythms in flies. So this is the same type of system as in this delayed negative feedback. It's negative feedback, but there's some delay. All right, we have two genes here that help control circadian rhythm in flies. Circadian rhythm, that's the day or night cycle, right? So if we shut off all the nights, eventually you get sleepy, or shut off all the lights, eventually you get sleepy, and eventually you wake up again, right? You have this kind of natural day-night type of cycle, this body rhythm, and the same thing with flies, they have that as well. So to control the day-night cycle in flies, you have two important genes. TIM gene stands for timeless, and PER as in period. So that's how you kind of remember circadian rhythms, timeless and period. Okay, so the timeless gene and the period gene both will be transcribed, so you'll get the RNA. The RNA will move from the nucleus to the cytosol. Eventually, mRNA gets translated. So here is the PER protein. Here is the TIM protein. Now, if it's daylight, or if you shine a light at the fly, what happens is the TIM protein will be degraded. So it's degraded in response to light. As long as it's daylight, as long as you shine a light, TIM gets destroyed. Right? You wake up the fly. PER protein goes around and does all sorts of different things. It says here regulated PER phosphorylation and degradation. Essentially, PER tells the rest of the cell it's daylight. Wake up. Right? PER says daylight time. Do your daylight stuff. Okay, so PER is affecting lots of proteins. Say, let's behave like it's daylight. Now, it's getting dark. So when it's dark, TIM will no longer be degraded. TIM and PER will form what's called a heterodimer. They'll bind to each other. After they stick to one another, they'll change a bit. Then they will dissociate and enter the nucleus. And PER will shut down expression of both TIM and PER. So this is a delayed negative feedback loop, as in you had PER, and it took a while, but eventually PER came back and you shut down TIM and PER. Took a while, but. Okay, so quick review, because I'll add another layer onto this. So TIM, timeless, PER, period. Both get transcribed. RNA moves from the nucleus to the cytosol. Here is the PER protein. Here is the TIM protein. If it's daylight, if you shine a light at the fly, TIM gets destroyed, it's gone. PER goes around telling the cell, hey guys, it's daylight. Behave like it's daylight. Now, if it gets dark, TIM will build up. TIM and PER will form a heterodimer. They'll kind of change a little bit. They will dissociate, they'll enter the nucleus. PER will then shut down both TIM expression and PER expression. Delayed negative feedback. Now, importantly, note, proteins do not last forever. Proteins don't last forever. So eventually, TIM protein, that's here, and the PER protein will be degraded. So eventually they stop working. Again, proteins, they don't last forever. Eventually, TIM protein and PER protein, they're going to be degraded. They're going to stop working. So the fly will eventually wake up again, right? You can put the fly in a dark room, it's going to wake up at some point. That's exactly what's happening. PER protein, TIM protein, eventually they get degraded, and eventually now you'll have more PER, right? And PER will go around and do daylight stuff, and you'll make more TIM, etc. The cycle continues. It goes over and over and over. So don't forget, these proteins do not last forever. Eventually, PER protein will be destroyed. Eventually, that fly is going to wake up. All right, quick clicker question. The above figure illustrates a complex transcriptional circuit. Which circuit is not indicated in the above rectangles? So buzz in if you can. Nice, yeah, good work. So C is the flip-flop device. So you can see a variety of, like you can see positive feedback, A going to B, going to C, going back to A, right? You can see the speed forward, B going to C, going to X, and then B skips forward to X, so it's a feed forward. And this is one of those kind of negative feedback or delayed negative feedbacks. D going to E, going to F, F comes back to D. Right? So kind of the last topic that we want to talk about is transcription attenuation. Both prokaryotes and eukaryotes have this premature termination of transcription. It's right up here, right? You need to write it. Okay, so it's called essentially what the title says. In transcription attenuation, right? The name kind of tells you what it does. Transcription, so affects transcription. Attenuation, as an RNA adopts a structure that interferes with RNA polymerase. Regulatory proteins can bind to RNA and interfere with attenuation. Examples include riboswitches, which function in prokaryotes, plants, and some fungi. So, riboswitch. Short RNA sequences that change conformation when bound to a small molecule are called riboswitches. So this is transcription attenuation. One example is a riboswitch. These are short RNA sequences that change conformation when bound by a small molecule. The example we're talking about is a prokaryotic riboswitch that regulates purine biosynthesis. So for this slide, it's the same deal if you took Bio 130 with me. You do not have to draw the different bases, but because we're talking about purine biosynthesis, you should remember which ones are purines, which ones are pyrimidines, and I gave you guys a really easy trick to remember this in Bio 130, right? So for pyrimidines, to remember that CT and U are pyrimidines, you see the pyramids, right? So you see the pyrimidines, right? So U, C, and T are pyrimidines. So everyone can remember that. It took you like 3 seconds. The other way to remember everything is a purine, or A and G are purines, is to think of someone with the initials A and G. So you think of something like Al Gore, that's who I use. So I think Al Gore stinks PU, and nothing wrong with Al Gore. I love Al Gore, right? He's amazing, but that's how I remember this. So Al Gore as an AG stinks PU, purines, right? So that's how I remember purines. So again, you see the pyramids for pyrimidines, and then Al Gore stinks purines, right? Al Gore stinks PU. And the other thing to remember is which one of these are single-ringed and which one of these are double-ringed. So pyrimidines are single rings, purines are double rings. And the way I remember this is I just think molecular biology is confusing, so the longer name goes with the shorter structure, and the shorter name goes with the longer structure. Okay, so that's how I remember. Okay, so it took you 5 seconds to explain this. Hopefully you all can remember this on the exam. Should not be a big deal. So, prokaryotic riboswitches that regulate purine biosynthesis, so A's and G's. Under low Guanine levels, transcription of purine biosynthetic genes is on. So what you have here is the DNA across the middle here. And you have here RNA polymerase transcribing the RNA. So here is the RNA, here's the 5' region of the RNA. And RNA polymerase here is going to be moving towards the right here, transcribing more and more and more. So here's the 3' end of the RNA down here, and it's going to add on to that RNA. It's going to add more and more and more as it transcribes more of that DNA. So adding lots and lots and lots and lots here. So transcription probably started way back here, right? It started way back here. RNA polymerase moved towards the right, transcribed a bit of RNA. It's going to keep on going. Now notice it started transcription before the genes for purine biosynthesis, right? So started transcription way up here. So this region, maybe up to about here, is what we call the untranslated region. So it's transcribed, but it's not translated. It doesn't contain coding sequences, but it's still important. So eventually, RNA polymerase will add on to that RNA, and you can imagine the start codon would be here, right? So that would be the start codon, and then the stop codon might be somewhere over here. Stop. All right, so to review the setup, here is the DNA. You started transcription over here. RNA polymerase is moving towards the right. It started transcription here. It's starting to transcribe lots and lots of RNA here. Now this region is what we call the untranslated region. Doesn't actually code for any protein, but it's still important. RNA polymerase is going to keep on going towards the right. It's going to keep on adding onto the RNA. Eventually on that RNA, you will get the start codon, right? So eventually it will transcribe that. Now this region up here, it doesn't code for anything, but it's called a riboswitch. So in the presence of high guanine levels, guanine will bind to the riboswitch. The riboswitch will undergo a conformational change and pull the RNA polymerase right off before it has a chance to continue on to the coding sequence. Right, so if you have high guanine levels around, well, you don't need to transcribe any more genes for purine biosynthesis. You already have lots of purine. You have lots of guanine around. So you have lots of guanine around. Guanine binds to that untranslated region, binds to that riboswitch. The riboswitch will change in conformation and pull RNA polymerase off. And so RNA polymerase never gets a chance to add on to that RNA to transcribe the gene, right? Now you can imagine back here, if you have low guanine levels, well, nothing binds to the riboswitch. RNA polymerase just keeps on going, eventually adds on to that RNA and adds on the start codon, the rest of the codons, and the stop codon. So just keep on going. Okay, so that is essentially the riboswitch. So with that being said, let's take a 10-minute break. I have 7:10.